Your phone vibrates when you get an email. So when I send an email, I can hear you got it. Hi, Gary.
Hi.
How are you?
Okay, how are you?
Not freaking out totally.
So, oh my God. I'm on the event page and the Hangout.
Cool.
And the event page shows me like, I don't know, half a minute ago.
Yeah, the YouTube feed is really slow. Hey,
So,
Gary.
hi.
How are you?
Wow. How are you?
So
Okay.
the YouTube feed is going to be like for my mom, and maybe if I send this to Penn, and other people, and then the Hangout's going to be for you and anybody else who really needs it.
Okay. Excuse me.
Um, okay, give me a minute. I just need to set up the screen mirroring. So I'm not going to, unfortunately, doing things this way, I don't think I can have like a preview, right? It's just the slides.
Uh, yeah, so what I did was to, share the desktop for
Mm-hmm.
that screen, so that way you could do the presentation mode, and then you see the full thing
I see.
there, right? So
if I do that,
that's probably what.
awesome. Now we're talking. And then I can go in here. Boom.
But yeah, let me join the handout and see what it's like.
Gary, you can see the slide, the gray slide?
Yeah. Mm-hmm.
Okay.
Yep.
Cool.
Okay.
Sweet. So that means I have presenter tools, and you're seeing what I want you to see. Okay, I'm just going to wait for Tomoki to join just so we can simulate a little bit of load.
Great.
And yeah, so I'm just, my goal in presenting now is, Gary, is just for you to see my overall flow of how I'm trying to present things. I'm not close to done, but I'm getting there. So, for example, I don't have an introduction. Gary, are you there?
Yeah, I am.
Okay.
Tell me when you're ready to go and I'll start watching, but right now I'm doing other stuff.
Okay. I think we're ready to go. Hello. Gary, I'm ready to go.
Okay. I'm coming back.
Okay.
Wait. I need to start taking notes. Okay. I'm ready when you are. Who else is there besides Tomoki?
Me and Tomoki, let's see, Akin and Yufei are busy at this time. They're going to look at the
Right.
recording that
Okay.
On Air is giving us. And I'll send that out as well. Okay. So, again, as a preface, I don't have an introduction. There's a bunch of, I don't have a conclusion. There's some stuff towards the end I haven't touched, and many of the slides aren't done. My goal is to show you the overall flow that I'm aiming for and the level at which I'm trying to present things, okay?
Well, don't oversell it.
Here's my first slide. I don't have an introduction.
Okay. Earth pigs? What?
Yeah. Okay, so I'm going to start. So thanks to Moki and Gary for coming. I've been in graduate school a long time, and I've had a lot of really great experiences, and I just want to start by reflecting on some of them. The first is our softball team, our championship softball team, the Earth Pigs. We were able to come together as a team and win I've also had the opportunity to go to Africa and meet a lot of new people in Africa. That was really kind of emotional experience for me, and I can really remember a lot of the faces of people I met there. And also just lots of discussions that I've had with people in my lab and people in the cognitive science department. I really appreciated all of that. Now, I study lateralization, and one thing I love about lateralization is it touches on every single one of those things I just told you about. The manual skill for playing baseball, or face recognition, even emotional processing, and of course, everybody knows language. Now, we know that brains are lateralized in humans from functional imaging, also from something called the water test. So if you inject the carotid artery with a barbiturate, let's say, you can hold on, what's the word? You can basically disable one of the hemispheres, and you can see that the person can still talk. If you disable the right hemisphere, they can't talk if you disable the left. And from split brain patients, where you cut the connections between the two hemispheres, and you can see them operating separately and see how they're different. Now, our lab studies vision, and so I'm interested in lateralization of vision. Great things about studying visual lateralization is that there's lots of neuroscience to tell you about how it might work. There's lots of computation to tell you what the principles are that build the visual representations internally. And we have good theories about where visual lateralization comes from. And also, it's just really cool, and it may work differently than you think. So here's basically a brain with two blue eyes, and he's looking at a fixation cross. And if he sees a stimulus on one side, on the left side of the screen, that stimulus will be conveyed originally to the right hemisphere. You get this crossover effect. And the same with the stimulus that's presented to the right visual field is communicated to the left hemisphere. But what happens if you do the normal thing and you look at something right in front of you, that stimulus actually gets cut in half, and half of the stimulus is sent to each hemisphere, and it's up to our brain at some point to combine that information and do the recognition and produce a response. And so what I'm going to talk about today is my work looking at generating a hypothesis about how the left and right hemispheres might differ in visual processing, and second, about how that communication might occur if that communication is important for visual lateralization. Right. So lateralization in vision, I haven't thought about this at all. So a number of stimuli are types of images are processed by the left and right hemispheres differently. For example, this larger, I flipped these, no I didn't. I see. I should probably.
Local level T versus global level T.
Yeah, that's not the problem. The problem is it's just flipped how I had it before. The global T is on the right because I put right hemisphere, not right visual field. Anyway, so whether you which hemisphere is better at processing the T, whether it's a small T, the left hemisphere is better at processing. The larger global level T on the right hemisphere is better at processing. If you present spatial frequency gratings and you ask people to classify them on a variety of different tasks, the left hemisphere will show an advantage for high frequency gratings, and the right hemisphere for low frequency gratings. And the left hemisphere shows more activation, better processing of words, whereas the right hemisphere in the same kind of area of the brain shows greater activation for face processing. So the current theory out there about how this happens is that it's all about frequencies, frequency filtering, frequency decomposition. The idea is that any stimulus can be represented as a frequency spectrogram. So this spectrogram represents the power inside that image, and I'll explain. So if you take a circle around this image, it represents a single frequency grading at a bunch of different orientations, and the color represents the power that is contained of that oriented grading in this image. As you go out to larger circles, you're incorporating higher frequencies. And all of these together, if you add them up, the power times the grading at that orientation, you will get this space. Now, another thing I can do, is I can take this two-dimensional representation of the frequencies and I can turn it into a one-dimensional representation of the frequencies. I just take the same grading at all different orientations and I average the power around that circle, and then I plot that at that frequency, the average power there at that frequency. And I can do that at each of the three frequencies I'm showing and build a one-dimensional representation of frequency versus power for this image. And so, in some sense, these are just three different representations of that same information that's in the image. So the current theory on what's different between the hemispheres is that the hemispheres process spatial frequencies differently. And here's a neural network representation of that theory, but it's really just a three-stage model. First, there's this frequency decomposition stage, which is like what I just showed you for breaking down the face image. And the second and third phases are filtering that frequency representation. So in the second one, you select a band where information is relevant for this task. So some part of that frequency range that you need to use to discriminate these images. And the second is a so-called attentional bias. An attentional bias where the left and right hemispheres actually differ. This is like top-down attention to the task. Now, the major questions for this theory are, how does this happen? Like, how does frequency decomposition happen? How does top-down attention affect frequency information in images? And second, why? Why would this happen? Where does this come from? Those are the questions I'm aiming to answer in my research. And so, again, a classic paradigm for showing these differences is this lateralized presentation, which I kind of already described here, where you flash an image, and that information is conveyed to the contralateral hemisphere. And if you flash the same image to each hemisphere, if you see different response times, differences in processing between left and right presentation, that can indicate that there's a difference between the hemispheres and how they process. And a classic test using this lateralized presentation paradigm is presenting these Nabon figures, where the subject's task is to press a button if you see a target letter like T. And so if the target is at the larger level, you'll see a faster reaction times if you present to the left visual field than the right. So an advantage for the right hemisphere processing. If the target letter T is at this local smaller level, then you'll see the opposite pattern. You see faster reaction times for right visual field or left hemisphere presentation. But it's not just, a lot of things actually modulate the asymmetry that you find when you present these stimuli. So the level of contrast in the stimulus will modulate it. Actually, the duration at which the stimulus is presented, the spacing of the letters will modulate the asymmetry. And even just the task. If you tell subjects, I don't want you to find the the target at any level, I want you to focus on the smaller level, or I want you to focus on the larger level. All of these will modulate the asymmetries. And so these are many of the same things that modulate what's called contour processing. This idea of an illusory contour, so this triangle here that doesn't actually appear, right? What you have are three oriented Pac-Men, but it looks to us like there's a triangle. And just in the same way, this larger level T is a illusory T. It's a contour, and whether you space things out more or whether you show them more quickly, that can modulate how strongly you see that contour. And so looking at a different kind of paradigm, here you can see this elliptical contour amongst many oriented lines. And when contrast is decreased, one way you can help determine that there's a contour there is to use information from your neighbors. So if you're not sure what the orientation of your within the receptive field here of your neuron, you can say, well, it's likely. Well, I talked to my neighbor and he said he's got something that's similar to me, right? And those are the green lines. And then I talked to my neighbors and they said they had something very different. This kind of, and we often see these kinds of curving lines. So actually, I think, I'm oriented in that correct way. So it's using neighbor information to inform about local orientation information. This is how people think contour processing is done. And so our hypothesis is that these kind of connections between neighboring receptive fields, neighboring neurons, may be different between the hemispheres, which drives contour processing differences between them. Okay. And so these are not your mom and dad's connections. Usually when we think of connections in the brain, we think of white matter connections, these things that we get through DTI, these fiber tracts that are just under the cortical surface. But instead, if you take that cortical surface and you inflate it and then you flatten it out, this cortex is just a really thin sheet. And if you inject some dye into that sheet, what you will see is patchy labeling. Around the right here, you'll see that at the injection point, you get a lot of labeling representing local connectivity within let's say a cortical column where most neurons there have the same receptive field. And then you'll see selective labeling of neighbors who have these invisible connections through the gray matter itself, and presumably and when you measure them, have different receptive fields. So this kind of neighbor communication that is used for things like determining contours under low contrast. And so if you take on the left here a retinotopic cortical area and a face is represented on it, you've got a number of these columns or neurons with different receptive fields that are selectively interconnected. And the way we think these neurons or these columns talk to each other is just like any other neural process. It just takes, this red guy just takes a weighted sum of his neighbors and then updates his own activation based on their activations. And that's exactly what this kind of two-dimensional neural network does on the right here. It just takes activation from itself and its neighbors and does a weighted sum to get its update. And we can just take that kind of hidden feature, that updated activation value, and we can project it back out to an image again and create what's called an autoencoder. This is a neural network which basically takes that feature and says, now I know how to go backwards and re-represent the image from that feature. And so you can learn what the weights should be, what the values of those weights should be, what that hidden feature should be, based on neural network training of a network like this. And so for a model like this, the only two parameters we need to know are the width of this Gaussian, so how wide is that distribution, and how many connections is each of those units making. Here, it's making five green connections, 5 neighbor connections, and 1 connection, of course, to itself. And yeah, so that's our model. We trained our model on a bunch of different data sets as follows. We create the network. So here I'm showing two hidden units out of 850. There's 850 input pixels, 850 output pixels, and 1 hidden unit per input and output. And the only thing we do is choose, again, the width of that distribution and the number of connections. And then just train the network on a set of images. So present the image, the network has an output, and then this downward arrow is seeing what the error is and changing the weights to try and do a better job of representing that image. You can do it again, and this time the image on top has less noise, and eventually it will say, I've done a good job of representing that image, and it will be done. And so after we've trained the network, we can, again, present another one of these images and then get this output image up here. We can do a frequency decomposition on that output image and see there's error in that image. In what spatial frequencies is the error contained in? We can also take these hidden representations, these feature level representations, which are affected by the connectivity of the network, and we can use them to do behavioral tasks, to do a classification task, just like we asked our human subjects to do. Okay. And so our hypothesis on what's different between the two hemispheres is that the left hemisphere has a wider connectivity than the right hemisphere. It connects to, the right hemisphere connects to nearer neighbors than the left hemisphere. We make this hypothesis for 2 reasons. One is there's only one asymmetry known in these kinds of connections, and it is of the width of connectivity. It's known in Wernicke's area, so an auditory area, where the left hemisphere patches connect to more distant neighbors than the right hemisphere. Now, this doesn't tell us about vision, but it tells us that this type of asymmetry is possible in the brain, and that at least one area has a connectivity pattern like this, so we would try it. A second reason to think so is that spatial correlations are greater at short distances. So if we think that the right hemisphere is better for contour processing, it might use shorter connections. And just to show that, when the neighbors are near each other, like on this eyebrow, right, you'll get a consistent contour over most faces. But if the connecting receptive fields are far apart, then you may get one in the eyebrow, but one inconsistently, maybe sometimes on the hairline, maybe sometimes on a wrinkle, maybe on anything else. And if you just map this out, if you map out every pair of neurons, you see that if your receptive field's at the center, it has a higher correlation to things that are nearby, so greater power in the center there. Gary.
Yeah.
You have the TV on?
What?
Is the TV on?
No, there's some feedback.
Okay.
Suddenly I started getting your picture with your voice. It's like your computer's mic got turned on or something.
Okay. Oh, I see. Okay. So
I no longer have a screen share now.
Okay. Let me set that up again. It's showing me that the screen share is still working.
Right, but it's showing me your picture with the circle around it as you talk.
Looks fine.
Can you
I mean,
click,
it wasn't doing that before.
can you click on my name in the Hangout?
Yeah.
Does that help?
No, it just gives me your picture.
Can you
Let me
click?
try. Let me try muting it. See what happens. Can you hear me now?
I can hear you.
That's not working.
Can you try clicking on? So there's two of me. One.
Hmm. Now I've lost your voice also. I muted you, but I can't seem to get you back, and I only have one version of you. I have the picture of you on Tomoki's. I've got, the video of you on Tomoki's, and
Yeah.
there you go. Okay. But I don't have the screen share.
You don't have the screen share?
Nope. Oh, it just came back.
Any idea what changed?
I clicked on screen share.
Weird. Okay.
And wait. No, it didn't come back. Hold on.
Sure.
No, that's my desktop.
Do you see three people in the Hangout?
Yeah,
And
I think I was seeing 4 before. I'm not sure.
okay. And you're clicked on the one of me that's blank, not the one of me that's an actual picture of me?
Yeah.
Mm,
Well, there's the video of you coming from Tomoki. There's
right.
3 people: me, Tomoki, and you.
Right. Okay. And you're
There
clicked on.
was there was Panchu before, briefly. Now he's gone.
Okay.
Can you try to turn the screen share off and on or something?
Yeah, I'm trying that.
Yeah, it's working from my end though. I can see the installation.
You see me, Gary?
Yeah.
Okay. And you see a blank desktop?
Nope. Yep. Now I do.
Okay. Let's continue.
Okay.
Now you see my slides, right?
Yes. Thank you.
Okay. So finally, I've got to the research. So we take this connectivity difference and we plug it into our model, where the left hemisphere model just has wider connectivity on average than the right hemisphere model. We train on these Nabon figures. There's 16 of them. There's LHTF at the lower and at the local and global levels. And after training, we take these hidden unit representations and we train a completely separate neural network to do the behavioral tasks that Sergeant asked our subjects. Give us a 1 if you see a target letter and a 0 if not. And we just want to see, do the left and right hemisphere networks perform differently on that classification task, indicating some biased representation from these connectivity differences. So here's the human data from Sergeant, and here's our model data based on that classification task. And you can see we have the same interaction between the hemisphere processing and the level of the target. And we can also take the left and right hemisphere images and do a spatial frequency decomposition and then compare how well each hemisphere is representing information at each spatial frequency. And so above the line essentially means the right hemisphere is doing a better job of representing that information. Below the line means left hemisphere is doing better. What you see here is at the lower spatial frequencies, the right hemisphere is representing information better, and at the higher frequencies, the left hemisphere is processing better. So this matches the experimental data from the literature and the existing model that both suggest this pattern of results. If we filter the images based on these spatial frequency differences, so filter the images and just keep the low frequency power, we get images that show a focus on the global level. And if we focus on the higher frequency images, you see it's keeping more information at the local level. And so this could be driving the difference, the behavioral difference we saw or the classification difference we saw between the networks. If instead of training the network on these Navon figures, we train the networks on faces, we see a similar preference for high frequencies in the right hemisphere model and low frequencies in the right hemisphere model and high frequencies in the left hemisphere model. And so just to summarize, we postulated that there's a difference in contour processing or long-range lateral connections between the two hemispheres. We implemented that as a sparse autoencoder with each autoencoder having a difference in the connectivity spread. And we saw that it has this expected association between wide connectivity, high frequency, and local level processing, and narrow connectivity, low frequency, and global level processing, all without ever representing frequency processing explicitly, like was done in the original model. We're postulating just an anatomical difference. Now, I've done a bunch of other work on this model, for example, presenting it with different tasks, so different stimuli doing different tasks where humans show asymmetries. For example, a face recognition task, you get a right hemisphere advantage, and the model can replicate those studies as well. I'm going to eliminate this and put that in an extra slide. Also, if we try to replicate...
Wait. Which are you going to eliminate?
The Chrisman one.
Oh.
I just don't have time to talk about it.
The one that doesn't work?
One that doesn't work.
Okay.
I'm just not even going to go there. Also, cortex is warped, so the representation of things at the center of your visual field, at the fovea, have a much greater representation in the cortex than things at the periphery. If we do that same kind of log polar warping of our images to be more like cortex, where this connectivity difference would appear, we still see this, and we also train on natural images. We still see the same frequency difference between the hemispheres, and we're able to, if we get the hidden unit encodings from networks trained on natural images, they also show the spatial frequency difference. And when we do behavioral tasks on these representations, we still see a nice, we still replicate a lot of the human data. Okay. I don't want to talk about any of those in great detail because I want to focus on how the model, let me think. Because I want to look at a study where I tried to corroborate this strange association between spread of connections and frequency processing. In the other experiments, I selected connections based on some distribution, some wide or narrow Gaussian distribution, and then I looked at what spatial frequencies were preferentially processed. The goal in this study was to select the spatial frequencies, train the networks on different spatial frequencies, and then allow the networks to choose the connections that do a better job of representing those spatial frequencies. So approach the problem from the opposite direction. So the basic methodology is start the left and right hemisphere, networks with the same connectivity pattern. So here's the connections for one hidden unit in each, but train them on different images. So in the left hemisphere, train them on the same images. On the right hemisphere network, train them on blurred, low-frequency versions, frequency filter versions of those images. And while training, remove the connections that are used the least in each network. And then after training, look and see how the connections differ. So you can see that they have, even though they started the same during this training and pruning process, they came out with different connections. And that's the study. The motivation behind this study is also developmental, because this type of study actually matches how visual development happens. So we know early in development that contrast sensitivity, visual acuity starts out poor. There's a bit. There are data suggesting that the right hemisphere starts maturing earlier than the left hemisphere, and so this indicates that the right hemisphere will mature while it has lower frequency information, lower visual acuity. The cortex matures both by strengthening connections and by pruning them. And so the right hemisphere will prune connections under blurrier, low-frequency input than the left hemisphere. Here's an example schedule of how, I haven't labeled this, of how the left and right hemispheres would, how they differ in their training. So on the top is the right hemisphere network, which is essentially starting to mature earlier, while the input images are blurrier, lower acuity, and the left hemisphere network starts maturing later when the images have started to improve. Even though it looks like the right hemisphere stops training, it actually continues training. Yeah. And so the question is, after training, what are the connection distribution different? How do the connection distributions differ between the hemispheres? What I've done here is I've taken all the hidden units for the right hemisphere network, and I placed them all in the center, and then I look at the distribution of connections that remain. I do the same thing for the left hemisphere network that started out with the same connections but trained them on full fidelity images instead of blurred images. And I subtract the two to compare. The warmer colors mean right hemisphere has more connections in that area. Blue means left hemisphere has more connections. And this just shows the same pattern of shorter connections for the right hemisphere trained on the blurry, low frequency information, and overrepresentation of long range connections in the left hemisphere trained on the full fidelity images. So this is the same pattern of results we saw in the first study, but approaching it from a completely different direction. I'm just going to skip this. Okay, and so again, I postulated a connection asymmetry in contour processing in long-range lateral connections, and then I implemented this as a sparse autoencoder, either with different spreads of connections or with different visual experience, and I saw that both approaches converged on the same answer, that these wider connection profiles led to a bias for higher frequency processing and local level processing, and the more narrow connections had a bias for low frequency processing and in global level stimuli. Okay, that's that part. So that was 30 minutes. A lot too long. Do you guys want to say anything now? Because I'm going to be shifting a lot off of this.
Okay, so that was actually 36 minutes.
Okay.
So now you're going to go on to new material?
Yeah.
Other material?
Mm-hmm.
I think you can, if you want to shorten this part, you probably can.
Uh-huh.
By, just saying having like one slide on development, for example, or
Mm-hmm.
etc.
Yeah.
I would, if you're going to leave it like that, I would put the visual development slide before doing the model.
Mm-hmm.
Okay.
Okay. So any clarification questions? I think I'm good. Okay, I'm just gonna keep going.
Okay.
So as I pointed out before, lateralization in vision is really interesting because when you look at something, half of it goes to each hemisphere. So ultimately, you really need to combine the two to recognize it. But what you're combining is different processing from each side. Excuse me. And so one question I had is, well, if I want to model central vision, right, I need two hemispheres that interact. What model do I use? Some models that have interacting hemispheres just treat intrahemispheric and interhemispheric connections the same. There's no time delays. They just combine information. Then there's a well-cited model that uses temporal delays because intrahemispheric connections tend to be shorter and faster than interhemispheric connections. And in this paper, they claim that this actually leads to a bias for intrahemispheric processing. So I originally got interested in callosal communication to try and figure out which of these models should I use to try and understand how central vision happens. But as I started to look into the literature, there was a lot of claims that the hemispheres may be relatively independent in humans. Again, when you do this WADA test, you can essentially disable one of the hemispheres. The other will still speak. The person will not claim that anything particularly strange is happening. The same is true of a split brain patient. So if you cut these callosal connections, and then you ask someone like JB, I believe his initials are, okay, so your corpus callosum's cut, your hemispheres are separated now. How does it feel? Is it any different than before? And you can go on YouTube and hear this guy. You can hear Alan Alda ask, do you feel any differently? And he says, no, just I have a backup brain now. Otherwise, all the same. And again, in this modeling work, there are some claims that the hemispheres process more independently because of the delays between them. So I needed to understand this to do my modeling. And the ultimate question boiled down to, does brain size affect lateralization? And I think the intuition is, in general, that it must. I mean, humans have big brains, we're lateralized, and so there must be some relationship between them. And there are two papers that really argue this more generally. One here, in this nice blank space, that argues as brains get bigger, the number of connections between the hemispheres is proportionally less. And so that decreases the amount of interhemispheric communication that you get. The other paper says that because the delays are longer between the hemispheres, you get less communication between them. Yeah. But as I looked into the literature, I'll just keep going. As I looked into the literature, a lot of questions arose. First, functional asymmetries are present in large brains and in small. So here's a functional asymmetry in, forget the name of the structure, in the frog. Second, humans without a corpus callosum, so very few connections or very, very, very slow connections, infinitely slow connections, don't show greater lateralization. They showed, in some cases, the same lateralization, in some cases, less lateralization. Third, even though delays decrease over development, the lateralization of language or of vision increases during development. So that doesn't fit the pattern. And lastly, interhemispheric functional connectivity is very strong during development and adulthood. So if you look at functional connections between the left and right hemispheres, corresponding areas, or between any random area, they're at least as strong and usually stronger than intrahemispheric functional connectivity. And so I wanted to look into what is the evidence. What is the evidence? So I'm going to go actually in turn. I have two more projects, one for each of those studies claiming less crosstalk between the hemispheres with bigger brain size. First looks at this fiber count. Okay. So these studies use something called allometry. And the basic idea here is that big brains are not big little brains. They are different. And the reason this has to happen is because you have some things that just increase with brain size, like delay. But you can't just have big creatures being slower to move than smaller creatures. You need to try and compensate to keep the overall behavior on the same scale. And to do that, different quantities have to scale differently with brain size. One good example is this surface area, or the cortical folding. You can see that in smaller brains, you have smoother, you can't see it too well, but you generally have smoother cortex. And as brain size gets bigger, you get more and more wrinkles. And so if you have a surface area, like the surface of the brain that's wrinkled here, and for a sphere, you would expect that surface area scales as 2/3 of the volume. Volume is r cubed, surface area is r squared, so you have 2/3. The actual is that it scales with 8/9. You get more surface area than you would expect for a sphere. That's where this wrinkling is coming from, and that's done to preserve connectivity distance and connectivity volume. These relationships tend to be power laws. So on Euclidean axes, you have that some quantity scales as like brain volume to some power, and the power can be less than 1, suggesting that what's on the x-axis is growing faster than what's on the y-axis. It can be linear, saying that they're both growing at the same rate, or it can be superlinear, suggesting that what's on the y-axis is growing faster. And for these allometric analyses, if you plot these on log-log axes, you'll get a straight line. You can do linear regression, and you'll see something with a slope less than 1, slope 1, or slope greater than 1. And that's it. That's everything I'm going to be doing here is linear regression on log-log plots. This is just to show you that these quantities appear in microscopic values, like synapses per neuron change with brain size, to macroscopic things like the cortical thickness, or even the number of areas that interconnect. Yeah, macroscopic quantities like that. And so this original study that claims less interhemispheric connectivity in larger brain animals took 11 primate species, MRI volumes of 11 primate species. They wanted to compare the total number of fibers in white matter fibers versus the total number of callosal fibers specifically. But from the MRI, what they were able to reconstruct as an estimate, what they were able to estimate and use as a proxy are gray matter surface area, which is like a proxy for the number of neurons in the brain, and the callosal surface area, which is like a proxy for the number of fibers in the corpus callosum. And when they compared these two values, so on the left is the Euclidean plot, and on the right is the log-log plot with a straight line, they found a power law relationship. What's different between these? Nothing. Okay. With the exponent of 0.88, less than 1, so what's on the gray axis, or what's on the x-axis grows faster than the y-axis. So gray matter area is growing faster than callosal area, suggesting that the number of neurons and the total number of connections grows faster than the number of callosal connections. And this is what they used to say that there's more independence between the hemispheres as brain size gets bigger. I think there's 2 reasons to doubt this interpretation. First, of course, they're using these surface areas as a proxy for connectivity, but the things that you need to compute connectivity from surface areas also change with brain size in not a straightforward way. You really need to include those quantities when you're doing this estimate. Second, I think their expectations were wrong because the scaling of inter-area connectivity differs between intrahemispheric connections and interhemispheric connections. Specifically, as brains get bigger, you have more of these cortical areas, and one area will connect to more of them. Whereas interhemispherically, you only ever connect in general to the cortical area on the other side, no matter what the brain size is. And so you should expect that the number of intrahemispheric inter-area connections and therefore fibers should be increasing compared to interhemispheric fibers. I'll draw this out a little bit more. So here's a small brain. It has 16 cortical areas, 8 per hemisphere, and here's a large brain with 32 cortical areas, 16 per hemisphere. And you can see that here each area connects to 4 other areas, and here each area connects to 8 other areas. That means that callosal connections here is 25%, right? And here is 12%. So it's decreasing with brain size. But I don't think this is the right measure because the same thing is happening for any of these intrahemispheric connections, right? This one here is still there, it's still just as strong, it's just kind of outnumbered. And so this isn't an intra versus inter effect, this is just an effect across all of these inter-area connections in the brain. And so I don't think that comparing the total number of connections is the right measure for functional connectivity. I think the right measure is comparing the number of fibers within one of these inter-area connections for an intrahemispheric versus interhemispheric one. When you control for the fact that they're expanding in one within the hemisphere and not across. And so, yeah. First, we have to compute the total number of fibers and the total number of callosal fibers, not estimate. And second, we have to compare. We have to see how this scaling compares to the scaling of the number of inter-area connections. Okay. Interesting. So here's the equations. They're really simple. The number of callosal connections is the callosal surface area times the density in surface area, number of fibers per unit surface area. And the number of white matter connections are the number of cortical neurons times the percent of them that project into the white matter. And the number of cortical neurons is just the gray matter volume times the neuron density. So actually, we have all of the quantities in the literature except one. The challenge I'm not going to talk about is really just pulling them out of the literature, which you have to do by parsing images and building a database. But when I do that, I'm able to plug in equations of how each quantity varies with total brain volume. Then I was able to do the same thing. I found a data set that contains 6 species, callosal axon density. I found that these look like, what do you call it, a power law, decreasing power law. When I plotted on when I grabbed these data points and plotted on a log-log axis and did the linear regression, I found that the fiber density decreases to the 0.28 minus 0.28 power, scales with the 0.28 minus 0.28 power of brain mass or brain volume, suggesting that as brains get bigger, you have less density in the corpus callosum. When I plotted the human data, which is not part of this data set, I got something that felt very close to the line, suggesting that the regression is holding for humans and is a good regression to use. So I just plug that value into this equation here. And when I compare callosal connections to white matter connections, I see that they scale with the 0.63 power. So just to go to the original, compared to the original estimate, here's what it looks like to scale to the 0.88. It's close to linear. Here's what it looks like to scale to the 0.63. It's something that dips much more quickly. And so the corpus callosum isn't keeping up with the intrahemispheric connectivity. Not even close. Why is that? So like I said, we have to compare. We have this confound of increasing number of inter-area connections. And so I do another regression. On the x-axis, I have the number of interhemispheric inter-area connections. On the y-axis, I take a ratio with the total number of interhemispheric, the total number of inter-area connections. On the y-axis, I do a similar ratio, but for fiber count instead, so the number of callosal fibers versus the total number of fibers. And when I compare those two, I find that they scale essentially isometrically. So this scaling, this relationship, and this relationship are the same, which suggests that all the changes to the fiber counts, to the ratio in the fiber count, is driven by the changes to the number of inter-area connections. I know that needs to be clear and shorter.
Ben,
Yeah?
that's the clearest I've ever seen it. Looks great.
Okay, thanks. I can just do a little algebra from those equations, from this last ratio. If I just do a little algebra from these quantities, I can compute the relative strength of interhemispheric connections, interhemispheric inter-area connections, and intrahemispheric inter-area connections. When I find that these fiber bundles across the corpus callosum are on average 4 times stronger than intrahemispheric fiber bundles or inter-area connections. And this is true even though on average it's believed that callosal connections are longer than intrahemispheric connections. Yeah, and there's whatever. Okay, so to summarize, Rilling and Insel suggested that there's this exponent of 0.88. The interhemispheric fibers mildly decrease with brain size, or the proportion of interhemispheric fibers mildly decrease with brain size. This suggests a functional decrease in callosal communication. What we found is that actually the interhemispheric fibers the proportion drastically decreases with brain size, but that this suggests no functional decrease in callosal communication. Whatever. It's driven by the homotopic nature of the callosal connections, whereas intrahemispheric connections are proliferating. Inter-area connections are proliferating. And a comparison of fiber counts between interconnecting areas suggests that callosal fiber bundles may be special. They're unexpectedly strong. And again, this is true across primates, including humans. Okay, so that's the first study. Didn't find any which didn't hold up to this idea that the hemispheres are more independent in larger brain species. The second one is this study looking at this neural network study, which looked at conduction delays. So the basic facts used in this study are that large brains have long axons, and long axons have long delays. Humans have big brains that are highly lateralized. And what they hypothesize is that due to the longer delays, you'll have less interaction between the hemispheres, and you'll have more interactions within the hemispheres where the layers are shorter. And you have less when you have this lesser interaction, you'll get lateralization. So let's just quickly see how this network works. So the network runs in time. receives an input here. Activation is propagated all through the network. All these connections that you see have a delay of 1 time step, except these interhemispheric connections, which can have a delay of 1 time step, so the same as the intrahemispheric ones, or 10 time steps, 10 times more. So time 1, you get some binary pattern on the input. That activation propagates forward over time and activates neurons. You can see on the right, time is increasing. It eventually reaches the output, and at some time later, whenever you want, while activation is bouncing around this network, you can just measure the output and then compare it to some expected output and train the weights of the network to try and produce that output at the end time 25. And so what we're going to do, or what they did in the study, is they manipulated the interhemispheric delay. It could be 1 or 10 time steps, and they varied the total amount of time that the network was allowed to run. And then they measured how these affect interhemispheric dependence, interhemispheric communication. The way they did that is they trained the network to produce these output patterns from the inputs to get them 100% right. And then they removed these callosal connections and just reran the network to see how well the network performed without the callosal connections. And the idea is that the amount of performance decrease that you see is related to the degree at which you needed those connections. And so it was communicating across those interhemispheric connections. So greater performance decrease on this lesion network means more interhemispheric communication. It needed them more. So here's how the network with delay 10, the long delay, in those interhemispheric connections, performs without the connections. And you can see initially it does very well at early stopping times, and then it drops off, indicating that at these times it wasn't really using those interhemispheric connections, because when they were missing, it performed fine. And you can see that's not the case for the delay equals 1 network. It was always using those connections, and so it has an earlier drop-off. So this is the evidence that delays affect interhemispheric communication. I wanted to see, if there's an onset difference, right, of 9 time steps. Is this just an onset difference, or is there additional independence between these 2 networks? And so I shifted one curve to match the onset of the other, and I looked for any difference between them, and I didn't find any. And so what this is actually showing is that the hemispheres coordinate, they talk just fine. It's just one's delayed in talking versus the other. There's no independence here. There's just a difference in the onset to interhemispheric communication. If you look on whether onset matters, you'll see maybe not. The magnitude of the differences used in this model are quite exaggerated. 10 times longer is not reasonable. In addition, the brain uses a distribution of fibers. There are, yes, very slow fibers in the corpus callosum, but there are also very fast fibers in the corpus callosum. And so an onset difference might not even be present if those fast fibers can be used to overcome this problem. And finally, as far as you think that the brain is a predictive organ, then these delays can be overcome by predictive activity sending that overcome those delays. I'm not going to go into detail. Okay. So this is strange because we know that distance matters. Like if you look at the connectivity of the brain, you can see that as the distance between pools of neurons increases, you get like a power law decrease with the number of neurons. You connect to things that are closer. That happens within a brain, like a across the whole brain in the macaque, but it also happens even if you just look at the corpus callosum. Those callosal fibers that are shorter in humans tend to connect more compared to the longer ones. So I just showed you two reasons why these long connections aren't depressed in large brains. Why is that? Well, the distance and this conduction delay both correlate with other things that matter. For example, volume. So one thing that I didn't show you before is that as brains get bigger, every quantity scales less than 1, right? The brain gets bigger faster than these quantities scale, except for one, and that's white matter volume. And this scales with the 4/3 power, which is essentially telling us that at some point in the growth of the brain, it will be overcome by white matter. It will be all white matter, because the white matter volume grows faster than the volume of the brain itself. So the volume, these long fibers have a volume problem. And that's one factor in why distance might matter. Another is metabolism. Long fibers can take up more energy. Finally, there's an issue of variability. If there's variability in that connection, in the communication of information that depends on distance, then you'll have a problem learning. Not even going to try. And so the question I asked is there any evidence of distance-dependent variability in the brain? And it turns out there is. And yeah, I'll give some references for that. By the way, this is the part where I haven't really finished. There is evidence that thin fibers, let's just do this, that thin unmyelinated axons have unreliable delays, and that the longer your thin unmyelinated axon, the more variability you'll have. And the time at which this is most important is in development. So you can see prenatally that the predominance of the axons are thin and unmyelinated, which are these white bars. As the brain, this is in a cat, after birth, you'll see that the axon diameters spread out, and so the proportion that are thin and unmyelinated decreases. And you'll see that eventually into adulthood, that these, in this black distribution, are the myelinated fibers. And so you'll have a lot of thick myelinated fibers and fewer thin unmyelinated fibers. This is saying that early in development, we have a lot of variability in conduction delays. What's the effect of that on interhemispheric communication? So I did a couple studies on the same similar neural network, the one I just showed you. Instead of varying the interhemispheric delays, I fixed them to 10 time steps, so some long time. And I implemented unreliability as delay-dependent activity and delay-dependent noise. So the longer the delay, the more the noise. The more active the unit, the more the noise. And I just compared a network with no noise and a network with noise to each other. And yeah. Oh boy. Okay, I think I'm done. I'm tired.
So how many people are you supposed to have?
I'm supposed to 45 minutes. I'm twice that.
Mm-hmm.
Not only that, but it's just too much.
No.
I'm exhausted.
My preference would be that you talk about this second two things and really zoom over the first stuff.
Are you talking about the Ringo stuff or just in general?
I'm talking about the fiber counts and the Ringo stuff, and not talk as much at all about the differential encoder model.
Holy crap. Okay. So you want to see me focus on the Rinsel and Ringo stuff.
Mm-hmm.
Okay. How much time should I spend on the differential encoding stuff?
Not very much.
5 minutes? 10 minutes?
Well, yeah, 5 or 10 minutes if you can do it.
I see. And then for this stuff, oh my God, it's so detailed, but I feel like it has to be.
Hmm.
I don't know. I'm okay with it. Any clearer? Is any of this stuff clear? Am I talking too much?
No, it's clear. The like I said, especially that the allometric scaling
Uh-huh.
was much clearer than I've ever seen it, and I really liked the visuals of what you're doing the ratio of.
Uh-huh. Okay.
So
Yeah. I agree. Well, I guess I have more comments on the first half part, but I guess you're going to be shortening it, right?
Yeah,
So,
but any comments is helpful. Will help me shorten, maybe.
so for the second part, what would be what's going to be like the punchline?
Yeah. The punchline for the second part is that interhemispheric communication is important in humans. That this idea of independence isn't there, and I really want to suggest that lateralization goes with interhemispheric communication, not independence. I'm very, very far from getting there, but at least I want to show that independence isn't there in either of these two papers that claim it.
Well, yeah. So maybe I don't know any suggestion, but
Sure.
maybe you can just start from that goal and then maybe try to reduce whatever details that may not take time.
Mm-hmm. I feel like I can give this talk like without any details and make a good story out of it. I'm just not sure how to. Like what I feel when I'm presenting is that the story's missing, which always happens with me, so that I can focus on the details and really walk through things. And I know how to tell a story without details, but the problem I don't know how to tell a story with details. Like I don't know how to do the two at the same time.
well, I think I don't know. I mean, because maybe it's because I've seen it already so many times, but I just think the differential encoder stuff, people may already know about.
That's not true.
Uh,
Yeah.
okay,
The other
well,
problem with
I
differential
guess that,
encoding. Go ahead.
yeah. I guess the other thing you can do is really skim over the details.
Yeah. That's my concern is that the details really matter.
Yeah.
They do.
Or you could just say, you've looked at this, and from here, my conclusion is such and such. Without necessarily justifying everything.
Right.
Because, yeah, it's all published and everything. So obviously, this time and time. So
Okay.
yeah, I could just maybe again, just go into details and explain like what your model does and what you have take a look at, but you don't necessarily have to justify like everyone's concept.
Mm-hmm. Do I just not talk about like the major takeaway for me from that what I really want to say is lateralization and visual processing is not about frequency filtering. It's about contour processing. And those are very different things, and those lead to very different predictions. I can't even get to their prediction stuff, but I at least want to say that they're very different things. And what I don't understand is, like, if I skim over things, I feel like I need to skim over the whole justification of how this relates to contour processing. Because if I skim over that, then the model can look like it's just a different way of doing frequency filtering. Because it seems to be doing frequency filtering.
Yeah, well, I mean, you have been making that connection,
Right.
So,
I need to make it shorter.
right. So what is the difference?
The difference is, the problem is I don't really show it in the model. But the difference is, if you do the differential encoding model, it shows spatial frequency differences, but that doesn't mean it's doing spatial frequency filtering, right? Like, it can take things that have differences in frequency content, and it can and a model that does frequency filtering might predict one thing, but a model of contour processing might say the contours there, even though the low frequencies aren't there, the contour's still there, and so it will show something different. Whereas if you filter out those frequencies, then that information isn't there. Even if the contours are there, that information, the low frequency information isn't there, so it'll show a different behavior. And there are tests to do that, and there's evidence in the literature that it's really doing contour processing, not spatial frequency filtering. But like I said, I haven't got there, both in the model and in the presentation. But that's the whole point of, for me, that's the whole point of a model. Is it's not about something abstract like frequency processing. It's about something concrete that we have a neural mechanism for, contour processing.
Yeah, well, I don't know. I mean, yeah, I think I like that presentation, that part of the presentation as it is. Anyway, so either, well, maybe you could mention the point on one slide and then say that's what you're getting to, or
Yeah, I
I
understand.
don't know. It might be fairly complicated. Right?
So complicated. Yeah. So, okay. I mean, like I said, I've just been working on slides and a lot of the visualizations and trying to explain things that I usually assume. I haven't really got to the presentation part and how it all fits together and how to use my time effectively. So,
Yeah, well, I think all the explanations are clear.
Okay.
Yeah, I think you're okay. So I guess I'll just say again what I said before, which is just, you know, you can have details on the slide, but you know that. Well, no, I think it's better not to have details on the slide, but be ready with details in like backup slides, which, you know, if you need them.
Uh-huh. Okay. So here's what I'm going to do. I'm going to start from scratch. And I'm going to make something simple. And then I'm going to add in details from what I have as it makes sense to tell the story and to back it up a little bit. And then anything that doesn't make it into there, I'll just put at the end as backup slides. I think that's a reasonable approach. Okay.
Say that again.
I said I'm just going to start from scratch.
No, don't start from scratch.
And I'm going to just listen. I'm going to start from scratch. I'm going to tell a really simple story in 10 to 15 slides. And then I'm going to try and pull in details to support that only, as needed. And then anything that I don't pull in that's a detail from the existing presentation goes into extra slides.
Right.
And that way I can be very clear about what my story is and then use the details that I've been working on to support it rather than be the focus.
Okay.
Okay.
Well, I just think you need obviously you're going to need another practice before Thursday, so hopefully we can, today is Tuesday, so that leaves tomorrow.
I hear you. I'm doing my best.
Okay.
I also need to not be dead by the time the presentation comes.
Right. I mean, this wasn't terrible. I thought it was a reasonable talk. You just need to cut it in half, and so, in order to do that efficiently, my suggestion was to cut out a large chunk of the talk.
Right.
And say, at the end you can say, I also did this.
Oh my God, now it goes to the end.
Or, something like that.
Okay. I'll figure it out. I'll have something for tomorrow, including an introduction.
Okay. Just for my information, though, what's wrong with thesis going over time? Is there any practical consequence when it goes to like an hour and a half talk?
Yeah, it pisses off your committee. That's not a joke. You don't want to do that.
Yeah, I mean, it's a public talk, so a bunch of people are going to be there, and they have short time. But also, it's like an hour for the public talk, and then it's supposed to be an hour for the private question asking. And I think if they don't have sufficient time to ask you questions, that may cause some, like your committee, may cause some problems.
Okay.
As well.
It's good to know.
Yeah.
So it's better to be on the shorter side.
Yeah. That's what I'm understanding. So my goal is 45 minutes. Okay, Gary, thank you. Sorry to keep you up so late.
I've been staying up late anyway.
And yeah, it'll be good if we figure out the Hangout stuff as well. Seems it went okay, but not 100% sure.
Yeah. Yeah.
So I'll practice that too. Okay, good night.
Okay. Good night.